everyone. I am Nolusindiso Williams. I am an intern from NMMU, Nelson Mandela Metropolitan University. And I will be giving you feedback on what was presented in the group academic research. The first speaker there was Lumka Dodana from the University of Rhodes. So now her oral presentation was a new biological control agent for the control of Pereschia aculeata in South Africa. This Pereschia aculeata is also known as lemongrass. It is found and it originates from Brazil and is also distributed here in South Africa. So it's found in the Eastern Cape, in KZN, in Gauteng, and some in Mpumalanga. But it is mostly widespread in the coastal areas, and I think more especially in Port St. John's. So now, how is this plant spread? This plant is spread by birds. So now the birds will carry the seed and they'll drop it off wherever, thus distributing it. But then why is it a problem? Why do we need a biological control for this plant? Well, it overwhelms our native plants because it's not from South Africa, so it uses up the resources that were meant for our native plants. So we will need to get rid of it. So now they've discovered a new biological control, which is called a Cathorinthia schneferia. Yes, schneferia. So this insect, it feeds on Pereschia aculeata. So that's why we need to use it, because it feeds on Pereschia aculeata, so it eliminates the problem. So the reason why we use this plant is because it is environmentally friendly. So it will not harm our plants, it will not harm our animals. And also it is sustainable and yes, it is safe. So now they will need to obtain this insect from Pereschia from Brazil. So they have to go to Brazil where the plant is originated from so that they can get the Pereschia aculeata specific insect. So they have to go all the way to Brazil to obtain it. Now, the aim of the study is to determine whether the agent is safe for the use in South Africa. So the methods that they used were they collected these plants, they collected different plants and with the major family of cactus. These plants were grown in a greenhouse and the experiments were done in an insect cage. And then all test species were replicated. And then 20 Pereschia aculeata were then also in a greenhouse, and they paired these plants. So each pair contained a control and each pair contained an experiment. So now the control was the one that was not treated and the experiment was treated with the insect. So now obviously now the result of that is the plants that were treated with the insect, they wilted and they didn't grow. And then the ones that were not treated, which are the control, they grew. So it shows us that now the insect fed on the Pereschia, which is a good result. And then so now they used different species of the Pereschia. So now they used Pereschia aculeata and then they used Pereschia gandifolia. Yes, they used that and they both treated those with the insect. And so and the rest of course. And then in both these particular Pereschia species, the insect developed into adult stage, right? They developed into adult stage, but then they didn't focus on the Pereschia gandifolia because Pereschia gandifolia is not found in South Africa, so we need not worry about that. So now we're just gonna focus on the aculeata. So now they compared these different species and they found that the insect developed in Pereschia, the survival percentage of this insect was 74%, so which is quite high. Yes. And then there was an interesting question posed this. The one person was concerned. They asked, they said, once now they are done with the insect and the insect has taken care of the problem, won't now the insect be problematic to us now? So the answer was no, because now there won't be any Pereschia aculeata. So if there isn't any Pereschia aculeata, the insect won't have anything to feed on, so it won't be a problem. So now what Lunga is striving for is that also they want now this insect to be released to be used as a new biological control agent in South Africa. And so now Lunga is going to continue doing her project, and she's going to be doing her master's in entomology. And now she's continuing with the same project, but now she's not going to use the Schaffneraya. Now she's going to use a new insect called the weevil. And the second oral presentation was by Megan Jacobs from the University of Cape Town. So now the topic is the psychosocial and biological effects of skin lighteners, with special reference to the influence of the media on the consumer's psyche. So, okay, firstly, what is a skin lightener? A skin lightener is skin lightening is application of chemical substances to inhibit melanin production. Melanin is the pigment that produces color on your skin. So now she touched on, okay, the skin lighteners have medical, they do have medical purposes, but now the media is now manipulating that in order to make people believe that lighter is beautiful. So now Hollywood stars like Michael Jackson and Rihanna and Beyonce, they all have this one thing in common. They use skin lighteners. And so now the media now influences us. So the skin lightening is rooted in women's psyche. And the self-concept, so now producing the self-concept that if now you are lighter, you are beautiful and you'll be more confident. You see, because now the whole point of wanting now because they see that they see these Hollywood stars looking lighter, and they see that they are confident, and then now they see themselves as which lowers their self-esteem, because now they're a bit darker than them. So now they also want to look like Beyonce and be lighter and more beautiful. So now, So now, okay, now the biological basis of this. So now the skin color is determined by the type and distribution and production of melanin. So now the skin lighteners, they block melanin production. So now the skin won't be, so now melanin protects us from UV. So now once now the production of melanin is blocked, then we won't be protected from UV light. So now the skin lighteners which are found in products are the hydroquinone and allicin. Hydroquinone, which has more adverse effects. So now the objectives of the study was to use descriptors advertisement to determine the use and the purchase of skin lighteners. And the second one is the effect of hydroquinone on skin cells. So now their method was they took different magazines. So they took a magazine from South Africa, which was Drum magazine, and then they took another magazine, I think from overseas, which was Ebony. And then they focused on the metaphors that were found in advertisements on those magazines, which are invoked. Okay, they use the metaphors in those magazines. No, because now they use subliminal languages to make you believe that having lighter skin is more beautiful and it is the in thing. So now both these magazines, they focus on good skin. That is, lighter and lovelier skin is more beautiful. And they also say that the more lighter you are, the more beautiful you are. And both say if you want to attract the opposite sex, use whatever cream that they are advertising, and you will get your life partner right on with your lighter skin. And so, and then now, okay, in their study, they used different years, right? Different eras and all that. So now, in the 1970s, now they started introducing technology. So now they'll be like, oh, science has discovered this, science has discovered that this technological method is much better and it is much safer to use. So now they use those things to manipulate the women's minds into getting these products. And then, like I said, the hydroquinone has severe effects on the skin cells. And in South Africa, they have the 82%, so 82% of the use of this hydroquinone causing a skin condition. So now what happens with the skin condition? Okay, what happens here is, now you buy this product and it has hydroquinone and it promises you that your skin will be lighter. So what happens now, you're gonna apply this lotion and at first, you're gonna seem lighter, and so now in order for you to sustain that lightness, you'll have to apply it all repeatedly. But then as you apply it, this hydroquinone will block the melanin production. So now the cells will more or less be dead. So now at the end of the day, later on in your late stage, now the product is no longer making you lighter, but it's making you darker. And now, and now the effects of this, now this is the terminal stage. The effects of these are not reversible. So now you will have that condition permanently with you. You will have those dark spots and all that. So, but then what can we do about this? Well, people need to be aware that these skin lighteners are not so innocent. So now we'll have to educate the industries, we'll have to educate the public, with public talks, and also use social media like Facebook and Twitter and what have you. And recently it has been discovered that more men are using these skin lighteners. So more men are using this skin lighteners. So it tells us that now this problem is expanding. It's expanding. So more reason why we need we need to have an awareness program of some sort, some organization of some sort. And also, hence, I was talking to Megan on our way back, and we were talking about how these skin lighteners, you can actually even find these products with skin lighteners, you can actually find them in outside. People are selling them in corners outside, and you can buy them with 5 rands and 10 rands and 15 rands. And these are the ones that have the illegal dose of hydroquinone. So yes, that was what we discussed. Thank you.
Hi everybody, my name is Paul. I've worked at Stellenbosch for the last year in an industrial biotech lab, and I'm going to talk to you about the academic industrial partnerships. We heard from Minister Hanekom earlier about the importance of applied research, especially to solve socioeconomic challenges in South Africa. I think these internships present a really great chance as a country to broaden our industrial impact, or the industrial impact of our science rather, and hopefully improve both our own lives through the opportunity we have received and others' lives through the work that we do and as the work kind of goes forward from now. I'm here to tell you briefly about the two academia industry presentations that we heard earlier. The first was on the decoloration of textile dyes with titanium dioxide nanoparticles and UV light. It was presented to us by Nangamso Nyangiwe at CPUT, and he was funded by TIA and the NRF for this work. The objectives of his research project was to investigate the efficiency of titanium dioxide nanoparticles as a photocatalyst to break down dyes with UV light. So what this means, titanium dioxide nanoparticles are very small particles that catalyze the breakdown of dyes that you find in the effluent from textile industry. It does this by producing free radicals from the UV light, and then those free radicals convert these dyes to carbon dioxide and water, resulting in a colorless product. In the textile industry, this would be applied because the effluent from the industry is actually quite toxic. It has a lot of solids and a lot of these so-called azo dyes. Conventionally, the industry has used membranes to achieve this decoloration, but this is expensive and time-consuming. So the proposal is to reduce the cost and the time for this treatment. His approach was to sonicate the effluent, which I believe decreases some of the other contaminants in the solution. Then immerse a UV lamp into this dye solution. Into the solution with both dyes and the titanium dioxide nanoparticles. Then leave it for a period of time, and then the sample is measured to see how much dye remains. He tested a range of dyes, and he showed that this approach allowed decoloration from anything between 30 minutes for one of the dyes up to 7 hours for complete decoloration. And this is much quicker than the process would be if they had used membranes. He also applied his technique to effluent from the textile industry rather than just dyes mixed into water. And this took a much longer time. This took 31 hours to achieve the same result of total decolorization. And this is mostly likely due to the high number of contaminants and other things in the effluent. So the future of this project, suggestions were that the titanium dioxide particles could be made smaller, which would make them more effective. Also, a higher power UV lamp could be used. His research used a 7-watt UV lamp or a 9-watt UV lamp, and it was suggested that it could go all the way up to 500 watts, which would obviously be a lot more effective in breaking down these dyes. And the project is definitely going big from here. There are already collaborations with other universities, as well as with the textile industries, and they're also in talks with the Water Research Council. The second project that we heard about, the second presentation, was on characterizing hybrid yeasts for white wine aroma. This was presented by Lucinda Christians, who worked at Wine Microbiology Unit at the Agricultural Research Council in Stellenbosch. She used a very common yeast called Saccharomyces cerevisiae. It's usually used as a beer yeast, but it's also used for white wines. And what's nice about this yeast is that different variants of the yeast give a wide variation in flavor. So it can obviously be used to enhance certain flavors and that kind of thing. The key part of the aroma and flavor from Saccharomyces cerevisiae are yeasts that release thiols. So these thiols are responsible for the aromas that we get. These thiol-releasing yeasts can also give off high volatile acids, but that is a problem. The volatile acids are responsible for vinegary tastes in the wine. So the objective of her work was to select and characterize yeasts looking for high thiol-releasing hybrids with low volatile acidity. She gathered yeasts from grape musts, which is the skins and stems and all the leftovers after squeezing the grapes. These yeasts were grown up in the lab and then characterized. She looked at protein content and types of proteins in the yeast, and then also performed a fermentation trial where various yeasts were used to actually make wines over a 3-month period. Some of the yeasts that she found showed very low volatile acids as well as good aromas, as evaluated by an independent sensory board. And that she also showed a large number of protein differences, which would be needed to be characterized further to actually understand the contribution they made to the flavor of the wine. The future of this project, she will be going on to an MSc to move the project forward, and also working with anchored yeasts to try and develop a hybrid that is lower in VAs, or volatile acids. Overall, both parties, both students mentioned that they experienced exposure to industry, which I think is very valuable. Nangamso in particular mentioned learned financial and project management skills, and both projects seem to have a good future because the research is applicable. And I think the nicest part was that they both are committed to taking the work forward and very excited about the work they're doing. For myself in this internship, I think I experienced or I learned a lot about the interactions between research and industry, and the state of the biotech industry in this country, and also just how it works, you know, how these meetings and interactions take the industry forward. Yeah, and that's it. Thank you very much to the speakers, to the NRF, and thank you for listening.
Good afternoon, everybody. My name is Ziphozakhe, and I am from Walter Sisulu University. I work at the Center for Global Health and Research. I'm going to be doing a report back on session C, which was research management, research support, and research administration. I think I should first start off with what our facilitator was talking about in the beginning, Dr. Kieran Dyson. He basically, the most important things that I kind of picked up from that was that research management and research administration is central to research. It is what facilitates the research. So it is a crucial part of research. And it is within both private, public, and semi-private research institutions. He also pointed out that within research, there are so many research administration facilities globally, and there's a society of these, and these are important research institutions because they assist in facilitating research, which is something that is essential. The reason why research is essential is that it's not only a job, but a necessity. It gives information on the biggest problems that we have in society today: the imploding human population, poverty, food insecurity, infrastructure, and economic activity. And the only way that these issues can be addressed is through knowledge, and that is through research. So without research, none of these problems can be solved. Information and research it is something that will create an environment where issues can be addressed. He made a clear example of Switzerland. Switzerland is one of the most powerful economies in Europe. And they don't necessarily have gold, or they don't necessarily have large production, or they don't necessarily have what other European economies have. But the one essential thing that they do have is knowledge. They have a research culture over there. And as a result, Switzerland has got the best living standards. Switzerland, who doesn't want to live in Switzerland? And that's because they just have research. If we would have research, or a research economy, or a knowledge economy, we would be able to address everything. So, moving on from that, I am going to start with the first presentation, and that was from Nopolo Mhlekwa, who's from Walter Sisulu University. And she was speaking about research management and research support and administration and her experiences through that. Yes. So the NRF internship for her was the first internship, the first job that she had. And it's obvious that in the beginning, like everybody else I'm sure, was very intimidated. And she says there were a little tears here and there in the beginning, and I'm sure, maybe myself, there were tears, but I'm sure other people also wanted to cry here and there. But from her experiences, she says that she has been exposed to research management and what research management actually is. This experience allowed her for a process of transition, to be able to see and experience how all of this happens. She said it was a transition because it's a reality syndrome. It's no longer school where you decide that, okay, I'm not going to go to class today, I'm going to bank. You actually have to be at work. You actually have to do the work that's allocated to you. She said that you actually had to learn and to be able to adapt to the situation. So it's adapt or die. With this experience, the effect of this is that she was able to learn interpersonal growth, to learn, she had inspirations, she started having a foundation, and she had something to wake up to in the morning. The goals that she learned is that she was able to motivate, inspire, and inspire, facilitate, and fund the people that are actually doing research, because researchers just do the research part, and the management has to be done by somebody else. It's like Santa's little elves. She was also able to represent the institution, because not only did she work in the NRF offices, she had to also work with students, so funding for students, for bursaries. She also was able to go through that process and learn that. And on top of that, she was able to recognize the impact of research in communities, because she was involved in planning workshops and involved in planning stuff that's involved with research that could actually help out the community or how it impacted on the community. The skills that she learned to be a team player.